Hello, I'm SBoyken. I'm a postdoc here in the Baker Lab. I did my PhD work at Iowa State University in Ames, Iowa. Yes, I did. I lived in Iowa most of my life until, pretty much all of my life until just about 3 years ago. What brought me to the Baker Lab is I was really excited about protein design. I wanted to be able to design allosteric and design specific interactions, and I really just fell in love with the idea of protein design. The project kind of started out as a side project within my first few weeks in the lab, and a lot of the design problems that we work on need specificity in a way that's really hard to design for, given how we typically design for proteins. I called it HBnet, hydrogen bond networks. What's special about it is that you can think of all hydrogen bonds in nature are really networks of hydrogen bonds. Proteins evolved and exist in aqueous buffer, water-based solutions, basically, where every single polar atom, polar meaning it has a partial charge, it's going to interact with the polar water atoms. And if a water gets pulled away, that polar atom has to get compensated by making a hydrogen bond, a polar interaction, to another protein atom or another biological polar atom. Once the linear polypeptide chain comes off the ribosome, it's going to be hydrogen bonding to waters. And so as the hydrophobic force, kind of greasy things that want to stick to each other rather than water, drives the protein to fold or two separate proteins to bind, then all those polar atoms, as water gets pulled away, have to make compensating hydrogen bonds to each other. And that's really what drives both protein folding and protein binding, is that you have this hydrophobic effect pulling the hydrophobic things away from water, and that drives the interaction. But then all these polar things have to come together in just the right geometry to make hydrogen bonds, which really restricts the conformation and really drives the specificity of the fold or the protein-protein interaction. To show an example of a hydrogen bond network, this is a beautiful design by one of my colleagues here, Posu, and you can see here the blue and white are hydrogen bond donors. So the white is actually a hydrogen, that's what gets donated, and the red are oxygen, which have a partial negative charge. And so you can think of these hydrogen bonds as a partially covalent, very geometrically restricted bond that really specifies this geometry. And you can see this network kind of branching through here. And all these white things are hydrophobic amino acids. So the hydrophobic amino acids are going to drive things together, but it's really the hydrogen bonds and the competing away of water that'll snap things into place and really lock it into a very specific conformation. Yeah, so the biggest challenge in finding networks is that it involves multiple residues. And for both computational efficiency as well as the types of interactions we can capture well when we're doing most protein prediction or protein design, involves pairwise interactions. So we have our design space, which you can think of as a pool of all the possible side chain conformations that we can design with. And typically, Rosetta is very good at taking those pairwise interactions and coming up with the energetically optimal solution. But hydrogen bond networks involve multiple residues. They're intrinsically not pairwise. And so it's a much more challenging problem that we have to search for these extensive networks where every polar atom that can makes a hydrogen bond. And it involves a lot of the same principles and a lot of the same design software underneath, but you can think of it as using it in a very different way. So we have this entire design space, and rather than letting the Rosetta energy function and somewhat randomized, highly efficient search algorithms converge on a sequence and conformation of rotamers, we extensively go into this design space and find all possible connectivities, networks of polar atoms, aka hydrogen bonds, and really find the ones that we like, by like meaning the ones that have the properties that we want, and stick them on as kind of seeds for design to really drive the design process to build the design around these hydrogen bond networks. Every polar atom that can make a hydrogen bond has to make a hydrogen bond, either to solvent or to each other. The hardest part is that when we're traversing this entire design space and finding all possible networks, we oftentimes get hundreds of thousands of networks, if not millions of networks, for a single backbone, a single scaffold. And so the hardest part is really picking out, of those millions of networks, what are the ones that we actually want, and which ones do we actually pick for downstream design to design the rest of the space and actually lead to something that we want to test in the lab. designing them in the same way that DNA uses them. Whereas in DNA, you have the double helix, and you have just these two hydrogen bonding interactions between either A and T or C and G, the Watson-Crick base pairs. And from those very simple set of hydrogen bonding interactions, we can design, and people in the field of DNA nanotechnology have been able to design a nearly unlimited set of specificities and also higher order assemblies and shapes in the field of DNA origami. We're actually doing them in coiled coils, which are structurally analogous to the double helix of DNA, which allows for these geometric cross sections that repeat. And so we can find the hydrogen bond network up here, we can repeat it down here, and we can achieve specificity in a very modular, very programmable fashion that is much like DNA, and that's something that's really new to protein design. The paper was definitely a very much team effort. There's a very talented graduate student, SBoyken, that we worked pretty closely on this. And Zibo and I designed a lot of these homo-oligomer coiled coils with lots of beautiful hydrogen bond networks. David was also a big part of the design process, especially in terms of the coiled coil backbones. Ben Groves, who's a floor below us in the CLIG lab, did the yeast-2 hybrid experiments. And there's a lot of other authors on the paper that contributed in very meaningful and useful ways. Being able to design new protein interaction specificities. We do a lot of very cool things with cellular engineering. Basically making new materials out of proteins from scratch, designing things that have moving parts, that have function. A protein that undergoes different conformations is switching between two specificities. And there might be a hydrogen bond network up here and a hydrogen bond network down here, and only one could be satisfied at a time. Then it could rock back and forth.